Well, here we are at key area 17 and it's looking at evolution. So first of all, have a look at the definition of evolution. This is what the SQA have got, and basically what they're saying is that it's a gradual change in the characteristics of a population of organisms over successive generations as a result of variation in the population's genomes. So what do they mean by that? Well, what they're doing or saying is that evolution is describing the changes that occur in a species, a particular species, over time. And that leads to offspring that are better adapted to survive in their environment than previous generations. Now, for evolution to happen, there's got to be changes that are taking part in the gene pool. This is where we've got this thing here about the population's genomes. What we're talking about here when we talk about the gene pool is, if it's a kind of odd term, but if you think, if we took a species and we thought of it, and you know I'm not very good at art, so this is a species of circle organisms, and they maybe have little legs. Some of the legs might be a little bit longer, some of them a little bit shorter. So if we think of all those organisms and we were to kind of gather them all together in this pool, obviously inside each of these organisms there are genes, and some of them have got slight differences than others. They're all little circular organisms, so when we're talking about a gene pool, we could be looking at a species of elephant or a species of snake or whatever, and you know that's our gene pool. And within there are the genes that that species have. Now, there are changes that can happen to these genes over a period of time, and there are different reasons why these changes take place. You've already had a wee look at mutations. So, I mean, if we think about a wee mutation that maybe takes place in this area here of this chromosome, and it has confers some kind of advantage, and so you've got natural selection there, and you've got that advantage there in this individual, then over time what will happen is that there will be changes in the frequencies of certain genetic sequences. So it would be over time that maybe the frequency of this gene that's here will start to increase because it confers some sort of advantage, as I've said. So you get this change in the frequencies of certain genetic sequences, or in other words, the alleles of a particular gene. Now, so evolution accounts for the origin of existing species from ancestors that lived long ago, and often the species that we have nowadays are very, very different from the ones that lived a long time ago. And we're going to have a look in particular at these areas that are here, because evolution involves obviously inheritance and selection. Natural selection's in there. We're going to look at something called sexual selection as well. And then we're going to look at genetic drift, and we're going to look at something called speciation. So these are the four areas that we're looking at under evolution. And so we're going to have 4 videos, and this is the first one here, and it's looking at inheritance. So inheritance is obviously the transfer of genetic material, but there's 2 ways that that can take place. One is called vertical transfer, and one is called horizontal transfer. So we'll have a look at what we mean by these terms. The first one, vertical transfer, that is where genes or sequences of protein coding DNA are transferred from parents down to their offspring, and that can happen either by sexual reproduction or by asexual reproduction. So obviously when we're talking about sexual reproduction, you've got 2 parents and they differ from one another genetically, and the offspring that come from them inherit different combinations of genes from each parent. When we're looking at asexual reproduction, we're looking at a single parent, and it produces an offspring which is identical to the parent. But both of these are termed vertical transfer. When we have a look at horizontal transfer of material, we're really looking at prokaryotes, that is bacteria, and there the genetic material can be transferred from one cell to another through horizontal gene transfer. So what we're talking about here is we've not got any sexual reproduction taking place. What here is we've got gene transfer happening where bits of DNA are transferred just directly from one bacteria to another, and that can happen in a number of ways. An example that I've got here is we've got something called transformation. You don't need to know these terms, but just kind of the general idea. And here it's when you've got cells or bacterial cells that are destroyed and bits of their DNA are sort of floating about nearby a bacterial cell, and another cell actually takes up some of that DNA. So here in this example here, you've got a bacterial cell that's breaking down, and here there's a little gene here that's got taken up, and it just happens to be an antibiotic resistant gene, and it gets taken up by this other bacterial cell, and it incorporates that bit of DNA into its genetic material. And bacteria are very adept at doing that kind of thing. It's why we end up with antibiotic bacteria. And there's this other process here called transduction, and it really comes when you've got a virus that replicates inside some sort of bacteria. Bacteria are actually infected by viruses, and here in this example here, you've got a little virus here that's infected this bacteria here. And what has happened is it's picked up a piece of DNA from there, and then it's gone on and it's come to another bacterial cell, and it's injected that DNA in, because that's what viruses do, and that DNA has been incorporated up into this bacterial cell here. Is this way here conjugation, and again you don't need to know these terms, but this one here is probably the one that the SQA is referring to in their document. Well, I've seen some of the questions around this kind of thing, and what is happening here is that an actual tube forms between two bacteria. It's called a conjugation tube, and what happens in this situation is that a plasmid from one bacteria is transferred into another bacteria, and bacteria do this in that way. They're swapping bits or giving bits of genetic information, and if you can imagine that here's a piece of genetic information that's got some antibiotic resistant gene on it, and it gets transferred to another bacteria, that helps to confer that antibiotic resistance to that other bacteria. So that is what we refer to as horizontal transfer. Really what you're doing is you're thinking about bits of DNA being transferred really from one cell to another. It's not involving any sexual reproduction taking place. Now, it's thought that the early evolution of prokaryotes, there was a lot of horizontal gene transfer, because obtaining a gene from a neighbour is much faster than waiting for one to evolve. So if you think even in just terms of antibiotic resistance, if you were to wait over time for certain types of bacteria to evolve that antibiotic resistance, it's going to take quite a long time. But hey, if your neighbour was to pass that gene to you, then that's a much faster way for evolution to take place. It's a wee bit of a risky strategy, as there's no guarantee that the genetic materials that is transferred is giving an advantage. It might actually be a disadvantage, but it's certainly one of the ways or one of the theories behind early evolution of prokaryotes. And I've mentioned already a significant amount of horizontal gene transfer already or still occurs in modern day prokaryotes, and the example that I've given already is the resistance to antibiotics occurring through the transfer of plasmids carrying antibiotic resistant genes from one bacteria to the other. Now, horizontal transfer of genetic material does occur in eukaryotes, that is animal cells. It's less common, but it does happen. And an example of that is where it is in plants. And really what is happening is it's gene transfer from a prokaryote. And what we're talking about here is a bacteria is called Agrobacterium tumefaciens. It's a bacteria that infects plant cells. And when it does that, the plasmid that it infects the plant cells with a plasmid, and that gets integrated into the actual genome of the plant. And we actually make use of this when we're doing genetic engineering of plants. We actually use this bacteria to get certain useful genes that we want to get into a plant. So that's one form of horizontal gene transfer in eukaryotes. Another example that we know all about is from viruses. So some viruses can actually integrate their DNA into the host genome, and they might remain there dormant as a provirus until they reproduce and destroy the cells. An example of that would be a herpes virus or an HIV virus. So the DNA actually ends up in the human's genome there, and that's an example of horizontal gene transfer in eukaryotes.